In 1970, K.H. Nielsen, T. Platt, and J. Woodland Hastings experimented with mutant form of Photobacterium fisheri strain MAV to understand the constraints that regulate the production of luciferase. Before getting into their observations, we need to understand some terms and certain concepts. So guys, you must be wondering, what is a luciferase? Luciferases are biological enzyme catalyst. It is produced in organisms like Renilla reniformis, Photiris lucicrescens, Omphallotus olearis, Echorea victoria, Vibrio havei, and Noctiluca scintillans. D-luciferin is the substrate of luciferase. Let's understand how is cell population and cell density related. So cell population refers to a collection of cells with typical proportions in peculiar phases of the cell cycle, whereas cell density refers to the number of living cells per unit volume. So you can see in the following diagram, cell population increases as the cell divides. With increase in cell population, cell density increases. Nielsen, Platt, and Hastings found that when the cells are in lag phase of the growth cycle, the gene that codes for luciferase production is not expressed, and therefore neither luciferase nor its messenger was being produced. But when the cell population enters exponential phase of the growth cycle, its cell density increases with the expression of luciferase gene. Increased cell density triggers diffusion of chemical molecules in the medium. These diffused chemical molecules are termed as autoinducers. Autoinducers are produced at the basal levels of the cells and they act as signaling molecules. The three scientists noticed that the occurrence of this phenomenon was free from the influence of any external conditions. In fact, medium in which the cells were suspended was conditioned in some way in the exponential phase when the cell density increased. They termed this phenomenon as autoinduction. What could be the significance of autoinducers and why would bacteria go for the production of autoinducers? Autoinducers are significant during bacterial communication. So bacteria can communicate by producing autoinducers. They respond to signals of autoinducers produced by other bacteria. Autoinducers can diffuse through the bacterial cell membrane. Therefore, their concentration inside the cell and outside the cell is approximately same. Upon reaching a critical concentration, autoinducers bind to the receptors in the bacterial cells. This results in the activation of those receptors. Activated receptors activate genes to express certain behaviors that help cells to respond to certain conditions in its environment, which may be beneficial or favorable for the cell. This event has been termed as quorum sensing. Quorum sensing is a cell density dependent phenomenon. Now, in the next 3 slides, you will find certain popular microorganisms who are known to produce autoinducers and how autoinducers are named. So, I have included those examples also against the name of the bacteria and what are their function, meaning what is the purpose of producing that autoinducer. So, I have included the functions also along with the name of the autoinducer and the microorganism that produces it. Thank you guys for watching my channel. See you in the next video. Till then, take care.